Again, I want to thank everybody for joining us for today's session entitled Unlocking the Full Potential of Your Single Cell and Spatial Genomics Research with Configurable LIMS. Today's session is sponsored by Astrix and will be presented by Sapio Sciences. Before we get into the session, you can go to the next slide, please. I just want to take a moment and share a little bit of information about who Astrix is. We put on these webinars on a very regular basis. We do about 2, 3, sometimes 4 per month. We oftentimes bring in a lot of our partners like Sapio. But who are we? We're a professional services company. We provide a wide array of services, everything from the selection and the implementation of laboratory informatics technologies through more strategic consulting, even through life science staffing services. We've been doing this for over 28 years globally. We operate from headquarters in New Jersey, but then we have centers of excellence in Central America and also offices throughout Europe. Over 90% of the employees that work for Astrix, over 700 strong, hold advanced science degrees. So we very much are a science-based company ourselves, helping other science-based companies. So with that, that's a little bit about who we are. So I'll take a moment now and introduce our speaker. So we can go to the next slide, please. And Dara McCreary is a scientific business analyst at Sapio. And as in this role, he leverages his PhD in medical science and extensive experience in the next generation sequencing to analyze and optimize scientific processes and workflows across the EMEA region. I don't have to obviously read Dara's rather impressive entire bio. So what I'd like to do at this point is turn the controls over to Dara, let him take you through the presentation, and I'll come back on at the end to help out with questions.
Thanks, Kevin. Yeah, as Kevin said, my name is Dara. I've been at Sapio for the last couple of years, working mainly as a scientific business analyst. And in today's presentation, what I'm going to go through is single cell and spatial genomic research using our Configurable LIMS platform. And I'm going to start out with an introduction to Sapio Sciences, then a brief description of both single cell and spatial genomics, and then look at some of the challenges that researchers have with that and how Sapio can really help. So first of all, I'm going to give an introduction to Sapio Sciences. We are a company that has a global presence. We have 5 offices around the world. We have 3 in America, 1 in the UK, and 1 in South Korea. And as Kevin mentioned, we have professional services working with us, such as Astrix. We also have Sapio resellers. So in total, we have 15 deployment partners. And what we really pride ourselves on is that we're one of the fastest growing lab informatics companies in the market. And we're trusted by 8 out of the top 10 pharmaceutical companies. So here's a brief history of Sapio. Sapio started in 2007 as a LIMS, and over time that platform expanded to include API enablement, cloud-based capabilities. And then in 2016, we added an ELN, which gave us a whole suite of tools that integrate with the LIMS in an all unified platform. And a hallmark of Sapio platform is the configurability, the no-code, the no-SQL approach, empowering users unparalleled flexibility. And in 2020, GHO invested in Sapio, which really accelerated our growth. And since then, as you can see here, since 2020, we really continue to innovate, introducing an AI-powered tools like Elaine and Jarvis as part of the scientific data cloud, and pushing the boundaries of what's possible with lab informatics. Our company's focus is to accelerate scientific discovery and lab operations. And we do that in many different areas, but some of them include biopharm R&D, clinical and diagnostics, and drug manufacture. In the biopharm, we have a multimodal discovery package which is easy to use, configurable for R&D with no code and built-in science-aware tools. In the clinical diagnostic space, we offer a streamlined end-to-end workflow from initial order, sessioning, sample processing, reporting to increase throughput, and is compliant with GLP and GCP. And within the drug manufacturing, we have a unified, flexible solution to maximize quality and compliance with GMP manufacturing environments. Sapio is a proven and trusted partner at scale. We accelerate scientific discovery by simplifying lab workflows. This is done by providing a unified platform that integrates all modules into a single seamless experience. So you don't have to go off and open a different tool. You can bring it into Sapio and bring your data there, and you can work in one platform experience. Where our platform stands out for being configurable and eliminating the need for coding and SQL expertise within your company. With built-in modules designed to work cohesively, Sapio delivers an all-in-one solution. So at a glance, Sapio delivers the first truly science-aware, AI-powered lab informatics platform, specially designed to simplify scientists' workflows and accelerate pipelines. One of the platform's many applications is the ease of use for experience and single place to access all your data. At the core of the platform is LANE, a natural language scientific AI interface that simplifies lab life, from querying data to designing plate layouts. The platform is highly flexible, again, featuring no-code or low-code capabilities, drag and drop workflows, no SQL data search, customized user interface, and robust rule engine. This enables the user to be able to do things themselves without having to go to the IT or the computer programmer in their company. It allows more flexibility, and smaller companies really to benefit from using Sapio. So the focus of today's presentation is to explore how Sapio Sciences can enhance single-cell and spatial genomics through our innovative LIMS platform. So I'm going to give you a short introduction on both single-cell and spatial genomics. Single-cell enables the study of individual cells, different cells within a complex tissue, offering unprecedented resolution in understanding cellular diversity, gene expression, and function. So using single cell, you can capture gene expression and epigenetics across thousands of cells, identify rare cell types and states. And this is really revolutionizing studies of cancer, neurobiology, and immunology. While spatial genomics integrates genomic data with spatial context, preserving tissue architecture and revealing how cells interact and organize in their native environment. You're able to map molecular signatures to tissue architecture, provide spatial insights into cellular interactions and microenvironments, vital for understanding development, disease progression, and tissue engineering, tissue repair. Sorry. So I'm going to focus on single-cell genomics. Single-cell genomics is a powerful technology that analyzes genetic material, either DNA or RNA, of individual cells. It allows us to understand the unique characteristics of each of those cells in a sample. And by uncovering the behavior and diversity of the individual cells within a tissue or organism, single-cell genomics revolutionizes our understanding of biological complexity. And there's key features of single-cell genomics, including single-cell resolution, capturing genetic differences between individual cells, highly sensitive, detecting rare cell types that might be missed in bulky sequencing, cellular diversity within tissues or tumors, identifying subpopulations and their functions. There's many applications for single-cell genomics, including disease research, understanding tumors evolution, identifying rare cancer cells. There's drug development, mapping cellular changes, cell fate decisions based off of drugs given. And development biology. Single-cell genomics helps track cell lineage, study cell differentiation, and understand embryonic development. And in immunology, we can study immune response profiling, immunotherapies, and look at autoimmune disorders. So if we're going to look closer at these applications, for capturing rare cell populations, rare cell types are events that can affect critical processes, either in cancer or immune responses. Bulky analysis, such as NGS for whole genome or or a whole exome can miss these rare signals, while single-cell approaches can detect and analyze them. Understanding cellular diversity. Human tissues aren't made up of diverse cell types that perform different functions. So single-cell genomics helps scientists define, catalog the full spectrum of cell types and states in healthy and diseased tissues. Disease mechanisms. Many diseases, especially complex ones like cancer, neurodegenerative disorders, and autoimmune diseases, are driven by changes in individual cells. So single-cell genomics provides a way of studying these changes in detail. And another thing that the area our scientific research is really going towards is precision medicine. So using single cell, we can look at biomarker discovery, identifying molecular markers for early disease detection or therapeutic targets, tailor-made treatments, helping develop personalized treatment plans on the cellular level, differences between patients. Spatial genomics is I'm going to discuss now. So what is spatial genomics? As I mentioned before, it integrates genomic data with spatial information, enabling the study of organization and function of genes within a tissue architecture. It shows genes are expressed, but where within the tissue they are active. Some of the key features is spatial context, so maintains the physical location of the cells within their native tissue, allowing for understanding and the context of cellular interactions and microenvironments. Higher resolution mapping, so we can map gene expression and other genomic features at a cellular and subcellular resolution. And this can be all combined in an omics science and multimodal omics science. Some of the applications, so disease research, understanding spatial patterns of gene expression, tracking how infections, autoimmune conditions affect tissue structure, developmental biology, cellular differentiation, tissue organization, regenerative medicine, mapping stem cell, allowing for better understanding how genes are involved in tissue repair and regeneration, and neuroscience, enabling the ability to explore spatial gene expressions in the brain regions to study neural circuits or disorders. A closer look at some of these. In developmental biology, cellular differentiation during development specializes during embryogenesis while mapping gene expression in early development. So the tissue organization, understanding how cells are working within a functional tissue is really important to understand. Regenerative medicine stem cell research, identifying spatial arrangements of stem cells within a tissue or their microenvironment, developing the understanding of how tissues engineer themselves via molecular cues, track how gene expression changes during tissue repair. And being able to understand this, we can potentially tap in and use that to our benefit. Disease research and precision medicine. So mapping gene expression across tumors, microenvironments is in cancer biology and tracking interactions between cancer cells and surrounding immune cells. Detecting spatial drug-resistant patterns within tumors and tissue-level immune responses during infection, mapping how pathogens invade tissues, interact with the host cells, linking gene expression patterns to specific regions to understand neurodegenerative diseases, such as Alzheimer's or Parkinson's, study how immune cells interact with tumors and respond to immunotherapies. While single cell and spatial genomics are transformative technologies, they come with their own set of challenges. So I'm going to go through some of the challenges and then go through some of the ways Sapio can help. So some of the key challenges is it's highly complex and the data volume that is produced. So on large scale, high dimensional data sets are complex to store and manage and analyze. And with single cell and spatial genomics, sample tracking is important to know where your samples are and where in the process they are. So managing and tracking thousands of individual cells through a sequencing process is prone to errors. Data quality and reproducibility is important. So noisy, dropout, batch effects compromises the accuracy and can compromise the reproducibility of your results. Data integrity, sequencing data with metadata and other biological layers with potential for comprehensive analysis and discovery. So the data is really key to understand and keep in a correct order so that we're getting the real potential from those results. And overcoming these challenges requires advanced technologies. If you're doing this by paper and pen or with Excel, it can really lead to problems later on. There are a number of areas that have bottlenecks within these workflows. So one would be your sample preparation, preparing thousands of cells at once, isolating individual cells, preserving spatial context without damaging the tissue. You really need to track all of this. During the sequencing process, you might be working on thousands of cells, and because of the high cost, time required to do all this tracking samples, you can get confused and lost and hard to track. And then obviously with both these workflows, the amount of data, the sheer volume, complexity generated can have significant challenges in storage and organization and retrieval. So effective data management systems are essential. And addressing these bottlenecks with improved workflows, technologies, and solutions that Sapio can offer will be key to fully realizing the potential of single cell and spatial genomics. So there's a number of strategies that can be taken on board to mitigate bottlenecks. So, community efforts are working together, collaborating, which is not always the easy thing to do in many labs today. Experimental design, designing the experiment up front, writing out all your protocols, making sure you follow everything can be tricky. Data analysis, bringing all your data from different workflows, from different instruments, all in one place. Sequencing efficiency, making sure that you have all the correct information, your QC data from your sequencing runs to ensure that you're really analyzing the best amount of data. Library preparation, so again, that goes with experimental design. There's many different steps, there's many different areas that things can go wrong, and tracking all of that can be difficult. And I've now realized I've gone the wrong way around, so I should have started with sample preparation. But again, sample preparation, similar. There's an ability to make sure you're doing everything to QC that the results at the end are correct. So I'm going to go through some of the benefits of using Sapio LIMS to enhance the lab operations and the data management. So I'll go into. So the Sapio platform, as I said previously, is a revolutionary platform designed to unlock your potential with your experiments. This cutting-edge platform provides a comprehensive suite of tools and features for analyzing, visualizing, interpreting data, and giving you better insights and discoveries into your own data. And how we do that? So Sapio has advanced algorithms and a user-friendly interface, enabling scientists to explore complex cellular landscapes with precision. The platform is tailored to meet the evolving needs of genomic research, enabling faster and more effective data analysis. And we do that through our no-code workflow customization. So drag and drop workflow builders to design, implement lab processes without programming expertise, allowing rapid adaptation or changes to your research protocols. End-to-end sample management, managing the complete life cycle of samples from reception to reporting, ensuring traceability throughout the entire process. Data contextualization and scalability, so facilitating comprehensive analysis, interpretation. The system is designed to scale. And with increased volume and complexity, you can do this using Sapio. There's an integration of advanced tools, so we seamlessly connect with various instruments and specialized systems to enhance laboratory capabilities and streamlining workflows. And there's also one of my favorite parts within this Sapio is the enhanced collaboration between members of your team. So no longer do you need to walk across the room or send an email and ask someone their results. Within the platform yourself, you can message your colleague and say, hey, I've got this result. I'm not sure what's going on here. Can you have a look? And you both can work through a workflow together at the same time, allowing you to interpret and explore and discover the intricacies in your results. So Sapio supports scientists, and we do that through seamless, streamlined sample management. So we provide a comprehensive sample tracking and management capabilities, ensuring effective handling of not just single cell samples from isolation to sequencing to reporting, but any different type of sample you might have. Enables researchers to monitor sample quality, track process steps, and maintain all the data linked to that sample throughout the experimental workflow. We've automated workflow management using customizable workflow templates, automates and standardize not just single cell genomic protocols, but many different workflows. This reduces manual errors, improves reproducibility, and accelerates sample processing times. Automating tasks, assignments, process tracking enhances overall efficiency within the lab. And there's integration with data analysis. Robust LIMS integrates seamlessly with bioinformatic pipelines, facilitating automated data processing and analysis. It provides a centralized platform for storing, organizing, analyzing, sequencing data, enabling researchers to quickly access and interpret results. So I mentioned that Sapio is customized streamlined workflows. The way Sapio has been designed, it has been designed to be flexible and adaptable. So it offers highly customized workflow templates that can be tailored to specific protocols. These are flexible, allowing researchers to adapt the processes and evolve as the cutting-edge techniques come out. Increase efficiency. So what Sapio will do is automate repetitive tasks, reduce manual entry, data entry, optimize resource allocation, and this leads to significant time savings, increasing sample throughput by improving overall lab productivity. Enhances data quality, so by enforcing standardized protocols, implementing automatic quality control checks, Sapio ensures that your quality across many different workflows is top-notch. This reduces the experimental variability and improves research outcomes, enabling and again improving collaboration, enabling seamless collaboration across teams with centralized communication, real-time updates, sharing access to data, and ensuring workflows are efficient and aligned. One of the things that Sapio does really well is instrument integration. This gets rid of the need for you to have USBs or external hard drives or dragging and dropping data between computers or emailing yourself results. There's automatic data transfer. Instruments integrated, automates the transfer of data from instruments to Sapio, eliminating those manual steps, which could be potentially produce errors. Streamlines downstream analysis, so data from instruments is readily accessible within the Sapio workflows for analysis, reporting, decision-making. Enhances sample tracking. So we're tracking samples throughout the workflow, ensuring traceability and reduces sample mix-up. So when you're doing your DNA extraction, we are producing the file that goes to the DNA extraction instrument and we are taking back the resulting file out of that, and then that can be linked to your QC data. So this is improving workflow efficiency, automate data transfer, sample tracking, data analysis. It significantly enhances workflow efficiency, saving time and resources. I think I've touched on it briefly, but we offer real-time tracking. So enhanced sample traceability. So enabling precision tracking of samples, ensuring that accurate samples are getting to where they need to be, and meeting audit trails for regulatory compliance. Increased operational efficiency, providing immediate feedback notifications, allowing quick issues, optimize workflow management to reduce bottlenecks. We also help with improving quality controls by monitoring samples, adhering to quality standards, and reducing errors in handling and analysis. I'll go back into enhanced collaboration and communication because I've touched on that several times. It facilitates also rapid decision making because all your data is in one place. All the information is in front of you, allowing for quick decisions based on the most up-to-date information, enabling labs to adapt protocols, strategies as needed. There's a streamlined data integration and analysis, ensuring that data capture is seamlessly integrated with analysis pipelines, simplifying the interpretation of results. Sapio is fully capable and able to support scalability of labs. So if you're a small lab and you're starting to grow, Sapio is able to adapt and grow with you. And they do this by efficient data management, so streamlined processes for handling and organizing data effectively. Does flexible workflows, so adaptable processes that can be customized to meet needs. Real-time monitoring, so you can continuously observe and track systems and processes. Robust integrations, so seamless connections, interactions with various systems and tools. And again, allowing teams to work together is probably one of the best features. Something that Sapio also does that is really good is the end-to-end workflow within Sapio. This isn't just genomic sequencing, I've used this as an example, but it includes from customer submission to project review, to sample management, to sample preparation, sequencing setup, sequencing run and monitoring, the processing, data reporting, and then return of reports. So this is really good for hospitals or people running clinical services, that they're able to bring samples in using our HCRM, and they can review all the samples within a project. They can track when a sample comes in through the sample management. They can then see the sample is moved to sample preparation, sequencing setup, so your library preparation, all of that is all tracked. And then once the sample is on the sequencer, you can actually track how the sample is processing through our run monitor capability. And once that is finished, you can then process the data, produce reports, and then send the report back via our HCRM. Sapio's HCRM is for healthcare and genomics. So it's a centralized data management to store, access patient data, sample data, research findings securely. It allows for a seamless collaboration, facilitating communication between the clients, clinicians, researchers, and patients, streamlining project coordination effortlessly. There's also the ability to look at your data and produce insightful analytics, track project progress, resource utilization, research outcomes, making data-driven decisions confidently. Sapio also offers the comprehensive reporting solution, so the ability to make tailored reports to meet specific project requirements with flexible templates for various stakeholders, automated reporting. So streamline reporting generation reduces the manual effort and minimizes errors. This has been put into place in some workflows like the NIPT workflow, where it's an end-to-end workflow within the Illumina instrument. And when the results are produced, we then are automatically able to make results or report results through the Sapio LIMS. Sapio also can build graphical representations to enhance data interpretation. It allows for all data in reports to be traceable, enhance credibility and reproducibility. So Sapio's end-to-end solution unifies customer relationship management using a centralized database for tracking customer interactions. And we have our laboratory workflow management to allow for sample tracking, monitoring samples from collection to analysis, ensuring that they are fully traceable. It also allows for customized lab workflows, tailors to lab processes to specific research needs and methods. And then finally, we have the ability to have comprehensive reporting, providing immediate access to results and metrics. And we also have the ability to automate customizable reports, enhancing data visualization and simplifying sharing of findings with stakeholders. What Sapio really is trying to do is to empower scientists. And what we're doing that with is data-driven insights, so allowing you to advance your understanding of complex biological systems. We're allowing for collaborative research, so collaborating with other researchers, sharing data, insights securely. And streamlining workflows to increase throughput, reduce manual errors, and accelerate research with automated processes. That's the end of my presentation.